Tonight's special guest speaker, Dr. Jennifer Sneed, is a research biologist at the Smithsonian Marine Station in Fort Pierce. Dr. Sneed holds a bachelor's degree from the University of Central Missouri, a master's degree from the University of South Florida, and a PhD from the Friedrich Schiller University in Germany. As a marine microbial chemical ecologist, Dr. Sneed studies all kinds of chemically mediated interactions in marine environments, specifically trying to understand the roles that marine microbes play in the ecological interactions of other organisms. Tonight, she's going to be telling us all about her research. Please help me welcome to the stage Dr. Jennifer Sneed.
Thank you, Zach, for that introduction. Let me get my water bottle situated. Can you all hear me? Am I good in mic? I'll try to stay here close to the microphone. So as Zach said, I am a research biologist at the Smithsonian Marine Station. So I'm gonna take another little poll. How many people here knew there was a Smithsonian in Fort Pierce? All right, well now the rest of you know as well. So we are actually a small marine lab, and we are sort of a satellite facility of the Natural History Museum in DC. So we fall under the Natural History Museum. And we were originally set up to really act as a place where visitors could come down from DC, people who were working at the Natural History Museum could come down and they could have access to the marine environments down here and be able to do their work, collect specimens, take them back to DC, that sort of thing. So we have a very robust visitor scientist program. We provide resources for visitors to come down and do their research. But we also have a couple of what I would call resident research programs. So one of those is the chemical ecology program, and that's the one that I'm part of, and I'll talk much more about tonight. And the other one is what we call our benthic ecology program, and they really focus on understanding the health of the lagoon by looking at the tiny critters that live in the sediment. And so they're very much focused also on the effects of freshwater releases from Lake Okeechobee and those sorts of things. So just a little overview of what we've got going on up in Fort Pierce. And if you're ever interested in coming up and visiting, we are going to start station tours again up there. We had stopped them during COVID, but we will have monthly tours where you can visit the research station up there. And then we also have a public aquarium. So how many people have ever been to the aquarium in Fort Pierce? Okay, keep your hand up if you knew that was run by the Smithsonian. So we actually, the county owns the aquarium, but we run the exhibit in the aquarium. So we're responsible for all of the actual exhibit materials, all of the aquariums, the different tanks, the different ecosystems that you see there. So I hope that you guys will make the trek up to Fort Pierce and visit us up there at the Smithsonian. So today, what I want to talk to you about, I'm glad that Zach kind of reiterated that tomorrow is Valentine's Day. I didn't even think about that, but we're going to talk a little bit about corals getting frisky. But I like to start out with this. Those of you who may be old enough to remember Madonna, I like to say we are living in a microbial world, and I am a microbial girl. So are you. All right, I don't know if I'm going to be the first one to break this to you, but you have more microbial cells in you and on you than you do human cells. You are mostly microbes. There are actually 100 trillion, approximately, microbial cells in your body compared to 30 trillion human cells. Yeah. And then when we look at that in terms of our genetic makeup, there are 99% of the genes in our bodies are microbial genes. We're only 1% human genetic material in our body. I told you, you're microbial girls and boys. And it's good that we have these microbes, right? I think a lot of times, and I think this is changing, so some of you might be familiar with some of this stuff, but in the past, I think a lot of times when we thought about bacteria and microbes and things, we just thought about negative things that make us sick, right? But now we're really learning that we have this whole microbial community in us and on us that really affects our health and how we even interact in the world. And these bacteria that we call our microbiome, that's what it's referred to as, the bacteria that live on us and in us, our microbiomes are really important in our health. And I'm just going to show you a few headlines that have popped up over the last 10 years or so of new things that they've discovered that these bacteria are doing in our bodies. So, for example, the bacteria in your gut may be helping you prevent you from getting kidney stones. There are gut bacteria that could affect your chances of getting Alzheimer's. So bacteria all the way down in your intestines or in your stomach are actually affecting things going on in your brain. Here's another example where they think that bacteria in the nose may be affecting asthma. And this is one of my favorites. They're actually starting to discover that we have bacteria in our guts that affect our mood and that can cause depression, or the lack of them can cause depression. So it's really fascinating to find out that we have these bacteria in us that really affect everything about us, our health, our mood, the way we interact in our environment. They're tiny, but they're mighty. And the way that these bacteria affect that, right? So how does a bacteria in your intestine affect your mood or whether or not you have depression? It's because they're producing chemical compounds. So in this study that I'm highlighting here, they found that patients who had lower numbers of bacteria in their guts that produce this compound called GABA were more likely to have depression. And GABA is an anti-inflammatory compound that's produced by bacteria. They also saw that people who had depression had lower amounts of bacteria that produce this compound called dopamine. Have you guys heard of dopamine before? That's one some people have heard of. It's a neurotransmitter, and it's a neurotransmitter that makes you feel good. And I really like this little cartoon that says serotonin and dopamine, technically the only two things you enjoy, because they are both feel-good neurotransmitters. So that's what makes you have that feel-good feeling in your brain. And so that's all controlled, or I won't say all controlled, but there are bacteria in your gut that can influence the production of these compounds that can influence your mood or whether you're depressed or not, which is really crazy. Okay, but I don't study humans. So I study coral reefs, and the same kind of concept applies to coral reefs. So when we look at this picture of this beautiful reef, we see a beautiful red fish, we see some corals, we might see some sea fans, some feather duster worms, like all these things. But what you're not seeing are the bacteria. In a reefscape like this, there are trillions of bacteria. So think about a teaspoon, the size of a teaspoon. In a teaspoon of seawater, there are 5 million bacterial cells. 5 million. So if you think about this water, I mean, that's a lot of bacteria. And I just told you how important these bacteria are in our bodies and in our guts. And the same thing is true for any ecosystem you look at, whether it be the seagrasses here in the lagoon, the mangroves, your garden, anything like that, and also, of course, on a coral reef. They're very important. And so when we look at corals, I'm going to do a little bit of a description of what a coral is to start with, because I'm not sure how familiar everyone is. But basically, a coral is an animal, and it grows in the ocean, obviously. But it's an animal that makes a skeleton, and it's attached to the bottom, so it doesn't move. And it also has these algae that live inside of it, so it's kind of like these tiny little plant cells that live inside of it. And those plant cells photosynthesize and provide food to the coral through photosynthesis. The coral can also eat. They can take in food from the water column, but they also get some of their food from the tiny algae cells that live inside them. And so when we talk about a coral, much like a human, I told you the human has their whole microbiome, a coral has its whole microbiome. And so we've started referring to corals as a holobiont. So it's not just the coral, it's the coral and its bacteria and its algae and its fungi and all of the different organisms that live inside the coral and on the coral to make it function. Just the same as us, really. And so you can see here that we have in this figure, bacteria, viruses are also important. This on the bottom where it says symbiodinium, those are the algae that live inside. Archaea are another sort of, it's very similar to bacteria. Endoliths, that's different kinds of algae and stuff that live on and in the corals. So I'm going to tell you 3 stories today. 3 stories about the ways that bacteria contribute to the health of a coral and to the coral ecosystem through the production of chemical compounds, because that's what I do. I'm a chemical ecologist, so I like to understand the chemical compounds that are being produced by organisms and how they influence their environment and the other organisms in the environment. So I'm going to start with reproduction. So I'll give you a minute to look at these little blobs. You guys see all these little brown blobs swimming around. As I zoom out in this picture, you'll see these are actually about the size of a fleck of pepper. So they're not very big, but you can see them with your naked eye. I just took that with my cell phone. And you can see they're in that's just a regular plastic mixing bowl that you would get at the grocery store or wherever. Those are coral larvae. So that is the juvenile stage of a coral. Before it's attached, it actually has a swimming stage. Just like a frog has a tadpole where the tadpole swims around, a coral has that little brown blob that you just saw. That brown blob, one of those will eventually grow into a coral like this. This is a coral called the mustard hill coral. Those were actually larvae from mustard hill corals that I showed in the last video. And then, of course, once you have many of those, you end up with a fully functional coral reef. But it all starts from that one little swimming brown blob. So just real quick to go over that, the coral reproductive cycle. So here's an adult coral, that's the mustard hill coral again. It releases these larvae, the swimming larvae that swim around that just look like little blobs of fat. What's basically what they are is little blobs of fat. But they do have cilia around them, so little hairs, so they're able to swim. They move down to the bottom. They find a good place to attach, and then they metamorphose. And that means they change shape. So in this photo, you can see on the left, one of those larvae that's still really long. And then right next to it, there's one that looks kind of like a little flower. That's what they look like when they metamorphose. When they attach, they change shape, and they kind of look like a little flower. And then hopefully, that will grow into a healthy adult coral, and the whole reproductive cycle will start again. So this stage where they move from this swimming larval stage to this settled metamorphose stage is really critical during the coral's life. I don't know how many of you guys have ever looked for a place to live in your life. I'm going to guess most of you. It's pretty important. You need to be in a good neighborhood. You need to know that it's safe. You need to know that you'll be protected. All of those things are true for these baby corals as well. I mean, I showed you their little blobs of fat. They're super tasty to anything that might come by. So they really have to be selective in where they attach on the bottom if they want to survive to be an adult. So amazingly, those little swimming bags of fat can make choices. And they make choices about where to attach on the bottom based on a lot of different factors. They can sometimes decide based on color. They can decide on texture. They can sense light. So when it's time for them to attach, they move away from the light and swim down to the bottom. They have a number of different ways they can detect their environment. But of course, the one that I'm interested in is chemicals. So they can sense the chemicals on the bottom and use those as well. And very interestingly, they are able to respond to very specific bacteria. So if you give the coral larvae a certain species of bacteria, they will attach and metamorphose. So they are actually attracted to certain bacteria. And what we've discovered is those bacteria are producing this compound called tetrabromoperol. And we shorten that to say TBP. It's much easier to say. So if you hear me say TBP, that's the same thing as tetrabromoperol. But this is a compound that's produced by these bacteria that the corals recognize. And when they sense that compound, they will attach and they will metamorphose. And so this is actually some work that just came out in publication last month. We've now tested this compound with 12 different species of Caribbean corals, and it induces settlement in every coral we test it with. And so when I say we test this compound, we put sterile seawater in a dish, so there's no bacteria, nothing in there. We add this compound, and they just attach all over the dish and metamorphose. So they are very happy with this compound. I just put this up here to show you that these corals are very diverse. So it's a broad range of families, genera, species of corals that respond to this, and even different life history strategies. It's very broadly applicable. And another thing about this compound that's really interesting, how many chemists are in the room? Just me? Okay. So for those of you who are non-chemists, you might look at all of these compounds that I've thrown up there, this alphabet soup, and think, those all kind of look the same, right? They don't look very different. Well, the truth is, if you were a chemist, you would think the same thing, because these compounds are very similar to each other. So the one I've got circled there, that's our winner, that's TBP, that's tetrabromoperol. That's the one that induces settlement. If you look at the one right next to it, can anybody see the difference between those two? It's missing one bromine. Br is bromine. It's missing one bromine. You take off that one bromine, the corals don't settle. They don't like it anymore. If you look at number 8 up there. Can you guys see the difference between that? It's got chlorine instead of bromine. Nope, they do not like it. Nope, do not care. So that is the only one of all of those compounds that they will settle in response to, even though a number of those compounds are produced by the same bacteria, but they only respond to the one very specific compound, which is very interesting. So what are we doing with this knowledge? I mean, obviously, I think it's fascinating because I'm a chemical ecologist, and I like to understand how organisms interact. But in a bigger context, we can use this knowledge to help with coral restoration. So we've teamed up with a institution, I don't know what to call them, Seacor International, and they do coral restoration through sexual reproduction. So a lot of coral restoration that you hear about is through asexual reproduction. So they basically fragment corals, which just means they break them, they let them grow, and then they put them out on the reef. But you don't get any genetic diversity that way, right? You're just getting clones of the corals you already have. So Seacor is really focused on getting the genetic diversity that you get from sexual reproduction. And they contacted us because part of their pipeline for getting new baby corals out on the reef is that they have to have that settlement stage that I told you about. That's so important, right? They have to get the corals to attach to their substrates and metamorphose, and then they can put them out on the reef. Well, what people typically do to get that to happen is take their tiles. You can see this star-looking tile. That's one of the ones that they use. And they'll put them out on a reef for a couple months to let a lot of bacteria and other things grow on it that the corals might like. But the problem with that is sometimes you get things that the corals don't like growing on there too. So they have to take them out, they bring them in, they kind of have to scrub off any fuzz that's growing on them to give a nice place for the corals to attach. And then sometimes the settlement's really spotty, so they'll attach, maybe one tile will have 100 corals on it, and then the next one will have none. And that's not really helpful, because when they want to put them out, they want all the tiles to have a few corals. Because you have too many, then they're fighting for space, and if you have ones that don't have any, then that's just a waste, right? So they contacted us to see if we could help with this problem. So we've been working with them on that, trying to use this compound, tetrabromoperol, to see if we could get more consistent and even settlement on these tiles. And we're still working on this. We've had some positive results, but this work is ongoing. And then we also got involved with this group on a project that's sponsored by the Department of Defense. So DARPA is the research arm of the Department of Defense, and a few years back they made a call for proposals for a program that they called reefs. And the idea of the reefs program is that they want researchers to develop hybrid artificial living reefs to put out along shorelines for shoreline protection. So basically, we know that the sea levels are rising, storms are becoming more intense. Can we do something to help protect our shorelines and improve the environment at the same time? So we got involved with this with a group from University of Miami. We call ourselves the X-Reefs team. This is our basic concept for the whole project. It's a really interesting project. It's got 21 different investigators involved, I think from 11 different institutions. It's a really huge project. The base of it is trying to develop a good artificial structure as the base for this reef, which involves a lot of things that I'm not familiar with, like materials science, what kind of concrete should we use, what kind of shape should that concrete be in, how many holes should it have, what's the optimum number of holes to make the waves crash the least, or all this sort of thing that's not in my wheelhouse at all. But that's what's kind of up there in that green area. And the goal that they set for us is to reduce the wave energy by 90%. So when a wave is coming to crash on land, we have to make sure that 90% of that energy from the wave never hits the shore, which is a pretty ambitious goal. The second area, which is in the blue down below, this is the ecosystem engineering part of the project. So this is the idea that they want us to build a functioning coral reef ecosystem on top of this concrete structure that we're going to put out there, which is a pretty ambitious task in itself. So we have to think about the kinds of corals that we're going to put out there, what kinds of herbivores we're going to put out there so that we can make sure we don't have too much algae growing on our reef, all kinds of different things going into that. And then this last part is what they're calling the adaptive biology part, and that's the orange corner up there. And that's how are we going to make this reef be sustainable into the future? Because we know sea temperatures are rising, pH is dropping in the water. We have all of these environmental stressors that are happening and that are going to get worse in the near future. So not only do we have to make a whole coral reef ecosystem, but we have to make it be able to withstand higher temperatures and all of these other stressors. So again, it's a very ambitious project, but it's been a lot of fun, and we've gotten to work with a lot of different people from different areas. So part of our part, so I just went through this whole part about how we can get corals to settle with this chemical compound. So this is one of our little parts of this project is assisted recruitment. So we're going to try to use that knowledge to see if we can enhance settlement on the reef or use it in other ways to enhance the coral cover on this reef as part of that ecosystem engineering of developing this coral reef. So that's one of our parts in this project. And then This I haven't really talked about much, but crustose coralline algae are these red algae that sometimes look like paint. So if you're at an aquarium or something, you see like a red purpley thing on the glass, that's usually crustose coralline algae. That's actually a type of seaweed that's growing on the glass. So corals really like to settle on crustose coralline algae. So that's another part of our project. Another one of our parts of the project is to try to help enhance the growth of these CCA on the reef to make it a better ecosystem for the corals. Okay, so that's reproduction. That's my reproduction story. I'm going to move away from Valentine's Day and coral sex to talk about some other ways that bacteria are important in coral reef ecosystems. This is a story about stony coral tissue loss disease. How many people have heard of stony coral tissue loss disease? Just a few. Wow, okay. So I'm glad I'm here to tell you about it. This has been a really devastating coral disease that hit Florida in around 2014 and then spread through our entire reef. I pulled this little infographic off the internet, and I've actually got some flyers in the back, especially for divers. If any of you are divers, it's some guides of recognizing stony coral tissue loss disease in the environment and also ways to keep from spreading it if you're diving. But basically, this is a disease that affects corals, and I think they've found it in 22 different coral species at this point, so it affects a lot of different corals. And it basically causes the tissue of the coral to die and fall off. So it really can pretty quickly kill an entire coral colony. And you can see in this, I've just blown up this little part where it says highly susceptible species, so you can see what these lesions kind of look like. So it causes these white lesions where the tissue dies and just completely falls off of the coral. And that's how it looks in a variety of different species. And so this is a graphic that shows how it spread across Florida over the years. So it started in the Miami-Dade area in 2014, and it has spread north and south, and you can see Martin County up there. Here's another quick quiz. How many people knew there were coral reefs off of Martin County? All right, a few more. Yeah, we have coral reefs out here. This is the northernmost area where corals are on the East Coast. Well, I should say tropical corals. There are some temperate corals that live up north, but we are the northernmost part of the Florida reef tract, and this disease spread all the way north through Martin County, and it's gone all the way down south. And this graphic actually ends in 2020, but we've actually found disease now down at the Dry Tortugas at the very end there as well. So it has spread through the entire Florida reef tract, and in fact now it's spread through most of the Caribbean as well. So another part of our part in the Reef Fence project is to look for probiotics. So I would bet most of you have heard about probiotics in terms of, maybe eating yogurt to help take in live bacterial cultures, or taking probiotic pills maybe after you've taken an antibiotic or something like that, again, to add helpful bacteria back to your gut. So we're trying to see if we can apply the same concept to corals. So can we find probiotic bacteria that will help the corals resist the disease or heal from the disease? And so as part of this project, we isolated many bacterial strains from a lot of different corals, and we started running assays to see if they were producing any kinds of antibiotic compounds. And the way that we do this is through this assay called a disc diffusion assay. So if you look at this plate, I hope you can see that there's kind of a fuzzy, blurry part all over the back of the plate, all over the plate. And that's what we call our target strain. So that's a type of potential pathogen. And so we want to see if any of these other bacteria can kill that pathogen. And so what we do is we spot little spots of different bacteria on top. And if there's a clear circle around those spots, it means it kept that other bacteria from growing. So we're basically kind of fighting two bacteria against each other and seeing who wins. So you can see here this one yellow one, MCH17, has a really big clear spot around it. So that got our attention. And we thought, well, let's see what's going on with this one. And it turns out that the compounds it's producing that are fighting against that other bacteria are this antibiotic called chloromycin, and then again, our tetrabromoperol. That TBP compound is actually not only makes coral settle, but it's also an antibiotic. So it produces both of these compounds, and we went ahead and tested this with corals, with live corals, to see if it would actually protect the corals. So this is a coral called Montastria cavernosa, and we just had different fragments of this coral that had disease, so you can see that kind of white disease lesion on the corals. This is actually two pieces from the same coral, so we've just cut that coral in half. We have two pieces, so it's exactly the same. And this is the before picture, and you can see the top one we didn't give any treatment to, and the bottom one we added our probiotic. And after some time, you can see when we had no treatment, that disease just moved across and killed most of that piece of coral. But when we added our probiotic, it didn't go any further. It stopped the lesion progression. So that was really positive. We were excited about that. We've moved these tests now to the field. So we actually go out into the field and apply the probiotic to corals out in the ocean. And this is how we do it. So this is my colleague, Kelly Pitts. She goes out and she puts these bags over top of the corals. And so the bag has a weighted line in the bottom so that it will stay over the coral and kind of keep a sealed area over the coral. And then she squirts the probiotics under the bag with that syringe. And then they leave it there for a couple hours so that the coral has time to take in the probiotics and hopefully get those bacteria inside of them. And then they go back and remove the bag. And then they go back and check later to see how the disease is progressing. So these kinds of assays are going on still. We have some sites in Broward County that we're studying. We have some sites down in the Keys that we're studying in a couple different coral species. And we have some promising results, especially from Broward County. Little different down in the Keys. There's a lot of different environmental factors down there. But we still think that this is a promising direction to look in. There are a lot of places that are treating these diseases with antibiotic paste, which seems to be helping as well. But just like in humans, there's concern about antibiotic resistance. So if we start treating corals with general antibiotics, not only are you more likely to you're more likely to get antibiotic-resistant bacteria, but you're also killing all the bacteria, right? The good ones and the bad ones, if you do that. So we think this could be a good alternative, or even maybe like adding the probiotic back after you've used the antibiotic. And then the last story I'm going to talk to you about today is about thermal tolerance, which is, can the corals withstand the water temperatures that we have today and that we're going to have into the future? And so I'm guessing that some of you heard about the mass bleaching event last summer. This was pretty bad. So these are just some of the headlines I pulled about this mass bleaching event. And you can see these are from CNN, PBS, so not even just local stories. These were national headlines. So we had a really bad bleaching event last summer. So not only have we been dealing with stony coral tissue loss disease for many years, now the temperatures are getting out of control and the corals just can't handle it. And unfortunately, some of the corals that were the hardest hit with the bleaching were ones that didn't get stony coral tissue loss disease. So they survived through stony coral tissue loss disease, but then they got hit with the temperature and many of them died. So just a little primer on what coral bleaching is. I told you that corals have these little algae that live inside of them. Coral bleaching is actually when the corals lose those algae. So it's not death. It doesn't mean they're dead. It just means they've lost those algae. And then when they don't have the algae is what makes them look that brown color. So when they don't have the algae, the tissue's clear, and you just see straight to the coral skeleton, which is why it looks white. The corals can take the algae back up. So if they have this heat stress event or some other sort of stressful event that causes them to lose their algae, and then that stress stops, they can take algae back in and they can move on with their lives. It is possible. But if that stress continues, it's often ends up being too much stress and they can't survive any longer because they're really dependent on those algae to provide them with food. And so you can kind of think like you're already stressed, then your food source goes away, and now you're trying to survive without it. It's pretty difficult. So they're not necessarily dead when they're bleached, but oftentimes they will die after bleaching. And this is just a photo that one of my colleagues took down in the Keys during that mass bleaching event. So all those bright white corals, those should be brown, and they were just all completely bright white. So we are trying to also see if we can find bacteria that might help corals resist these increased temperatures. So that's another big part of our portion of the Reefs project, is to try to help enhance thermal tolerance through the addition of probiotic bacteria, which seems like a pretty crazy idea, but we're going to go for it, and there's been some other researchers who've had success with this sort of thing. So actually what we start with is just doing some laboratory screening, right? Our goal is to screen 500 different bacterial strains from 4 different coral species. We can't do that with corals. Like, we can't test those strains against live corals that we would be decimating a whole reef to get that many corals to try to test them with. That's not a good idea. So we do some lab-based assays to look for characteristics of those bacteria that we think might be beneficial to corals, and then we'll try them with the corals. So what we're looking for are iron supplements, antioxidants, and vitamin B12. So again, you might be thinking, this sounds like my medicine cabinet. It's very similar. So these are all things that can really help the algae that live inside the coral be happy. We want to keep them happy so they don't leave and they stay in the coral and provide their function there. And so this assay on the right, you can see these plates that are a little bit blue. This is an assay looking for bacteria that are able to take up iron. So if they can take up iron from the environment and provide them to the algae, that's a really huge benefit to the algae, because the algae need the iron to grow, and it's a limited resource in the ocean. So the way this assay works is we have this blue media. If we plate our bacteria on there and the media changes color, if it turns yellow, then we know it's taking up iron. So when we look at these plates, you can kind of see the bacteria are the darker yellow kind of spots. That's actually just the color of the bacteria. But if you look around them, you see a kind of a halo effect. And that's the indicator that they're taking up iron, and that these are ones we might want to look into more. This other assay on the bottom, this is an antioxidant assay, and this is actually used a lot in food science. So when people are looking at different agricultural products and trying to figure out what's producing antioxidants for people to eat, they do these same assays. So this is also a color-based assay. So if the color on this plate is purple, then antioxidants are not being produced. But if the color changes to yellow, then the bacteria is producing antioxidants. So we're running these 2 assays. And then we're also doing a genetic-based assay where we actually look for genes in the bacteria that we know produce vitamin B12. And so those are the 3 assays that we're running. We'll screen all 500 bacterial strains that we have and see which ones seem to be the most promising in terms of being able to take in iron, produce antioxidants, and produce vitamin B12. And then we'll go ahead and move on to testing them with live corals. And this was a pilot test that we did. This is a staghorn coral, Acroporus cervicornis. This was a temperature experiment. So the one on the left this was before we increased the temperature. You can see it's nice and brown and looks good and healthy. And then as we increase the temperature after 9 days, this one not only did it bleach, but it also died. It lost all of its tissue. So it did not like it. We tested a number of different probiotics, potential probiotics in this assay, and we didn't have any that really helped the corals at all. So we're gonna work a little harder on our screening assays and see what we can find. So our hope is that eventually, one day, we can get back from these decimated reefs that we have out there right now, back to these healthy, vibrant soups of bacteria that we know and love. And with that, I just want to leave you with the impression that microbes rule the world. They rule our bodies, they rule our environments. They're really in charge of everything. I want to go ahead and open it up for questions and just let you know that the Smithsonian is partially federally funded, but we're also partially donation funded. So if you're interested in this work and you're interested in supporting it any further, you can contact me, or you can go online to make donations, or you can send a letter to the Smithsonian Marine Station any way you want, or we can just chat afterwards. Any of that. So, do you guys have any questions for me? Oh, you can also scan the QR code. And if you need help with that, I can help you with that. Sure.
Are you also collaborating with like Dr. Voss, Grant?
I don't actually collaborate a whole lot with Dr. Voss, but there are two teams who were awarded grants by DARPA. Well, there's three teams. There's two coral teams and an oyster reef team that were awarded DARPA grants, and he's on the other team. Mm-hmm. Yeah.
One diagram there is stony coral, stony in the Miami area is spreading. Is there a cause? Did you find out what the cause might be coming from a metropolis like that?
They don't know still. Oh, I'm sorry. Thank you for reminding me. He asked me to repeat the question. So the first question was I collaborating with Dr. Josh Voss at FAU? The second question is, do we know the cause? It looks like the stony coral tissue loss disease started near Miami-Dade and has spread from there, and do we know the cause? And we don't yet really know the cause. So there's been a ton of research going on trying to understand that. There's some thought that it may be related to some dredging that happened that stirred up sediment at that time, but it's really not fully understood. There's some thought that it's actually a disease of the tiny algae that live inside the corals, so that they're the ones that are actually getting diseased and it's killing the coral, that's how it's brought into the coral. But it's still a bit of a mystery, and there's still a lot of intense ongoing research. There's a whole group across the state that is funded by the Florida Department of Environmental Protection, who meets on bi-weekly calls to discuss all of the research going on across the state about stony coral tissue loss disease. Yeah.
Just kind of a follow-up to that. I know it's not your specialty, can you comment on current thinking at physiology of stony coral tissue loss? I know it obviously must be bacterial because of the stones and antibiotics, but I understood there may be a viral component which is linked to it.
Yeah, so the question is sort of what kind of organism is causing it. So again, it's not fully known, and that we've done a lot of work at our lab trying to figure that out. We do have an indication that it may be a bacterial cause because treating it with antibiotics seems to help. But it could also be that there's some underlying initial cause, and then there are other opportunistic bacteria that are coming in and causing a lot of the tissue loss and things. So that it's not really the bacteria that are the initial cause, but because they're causing a secondary infection, that's why the antibiotics are helping. So we've done quite a bit of work trying to figure out if maybe we could separate out whether it was viral or bacterial. Again, it's still all ongoing. It's not really known yet. Yes, Zach?
So we've got some great questions coming in from our Zoom audience, so I want to grab a couple of these. The first one is, how did you isolate TBP from the bacteria?
Trying to decide if I should show you guys all my slides that are down the stack. I will give you a very brief guide to how chemical ecologists discover compounds. So we do a process called bioassay-guided fractionation. And what that means is when we're looking for a compound, so initially I had tested 25 different bacterial strains by themselves, and I found one that the corals like to settle on. And so when I knew I had this one bacteria that they liked, I extracted all the chemical compounds from that bacteria, and then I gave them just that whole big soup of bacterial compounds. Yeah, so it's like that could be hundreds of different compounds in there. I just tested that, and they settled in response to that. And once you know they're going to settle in response to your mixture of all your different compounds, you can start to break that mixture into parts, right? So you can think of it like let's say I had 100 compounds, and I broke it into 4 parts, and we imagine like each part had 25 compounds. And then I tested those 4 parts, and only one of them they responded to, right? So then I get rid of those other 75 compounds, now I'm down to 25. And then I broke that into like 10 parts. And then I tested those 10 parts, and then they only responded to one of those 10 parts. And so we continually go through that process until we get to one compound, if it is one compound. And then we have different methods for identifying that compound once we have it isolated.
All right, next question is about the concrete hybrid reef designs you mentioned. Have they been tested in the wild yet?
Oh man, we're working on it. So that project is actually headed by University of Miami. And they have, as part of a different project, had already had what they refer to as the test bed, as sort of some artificial reef structures that they've put off the coast of Miami. So we're able to use that to test out some of our strategies. The first deployment of our actual structure will go in the water, I believe it's May of 2025. Oh, I hope that's right. It's sometime in the next year and a half. It's the first deployment. So we're supposed to deploy, it keeps changing. I can't remember now if we're doing 2 deployments or not. But we'll be deploying the structure, and then we'll be doing a bunch of tests on the structure of all of the different experiments for the ecosystem and the thermal tolerance assays and that kind of stuff we will do out in the field.
Our next Zoom question kind of gets to the heart of the so what when it relates to coral restoration. And the question is a big one. What's the environmental impact of coral death?
Okay, yeah. So the environmental impact of coral death, so basically the question is, what do coral reefs do for us, right, is the basic question. They are one of the most biodiverse ecosystems on the planet. So for one thing, if we lose our coral reef ecosystems, we'll lose a huge amount of biodiversity. I didn't go into this in this talk, but one of the other kind of flip sides of chemical ecology is what we call natural products chemistry, which is focused on looking at the compounds that organisms produce, like TBP, and seeing if they have any pharmaceutical applications for humans. So we, in our lab, that's another thing that we do. We isolate some of these compounds and we send them to collaborators at University of Florida who see if they have anti-cancer activity. So one of the things we always like to point out when we're talking about biodiversity is that there are potential life-saving drugs in the sea that we haven't found yet. And if those organisms go extinct before we ever find them, then we're never gonna have those drugs, right? So that's one thing. That's just one small reason that biodiversity is important. And again, we talked about this project that they're asking us to develop a living coral reef to protect our shorelines. That's because coral reefs protect our shorelines. So when they die, we lose that shoreline protection, and we know we're having this increased the sea level rise, increased intensity of storms. You know, places like Miami especially are interested in this kind of research because if they lose that protection, it's really going to be devastating to those coastal cities, coastal environments. Economically, there's a lot of places that rely on the money that comes in from tourism from from coral reefs, and a lot of island nations depend on them for food because they're a hotbed of a lot of different fishes and things, and if you don't have the coral reef there, the fish won't be there, and that food source will be gone. So those are just a few examples of why it's really important to try as best we can to keep our coral reefs happy, functional, and here, basically. So here.
I'm wondering, I'm sure you've heard about the coral reef which was suddenly reproducing in the south of Australia, south of the reef which died further north. No?
It was suddenly reproducing?
Yeah, it suddenly reproduced, and they have a wonderful coral reef south of the reef which they lost.
So there wasn't a coral reef there before?
No.
Oh, I see. I'm not familiar with that. So the question was about a reef in Australia where they had a devastating event to a reef that wiped out the reef, and then there was a new coral reef that kind of popped up south of that. I'm not familiar with that. I'm not sure. There was another question back there?
Yes, can you speak at all about the health of the barrier reef off of Andros? I mean, I'm sure this 2023 bleaching event very much affected the health of the reef there. I'm just curious if you know much about the Andros reef.
Okay, so the question is whether I know much about the health of the reef off Andros. I honestly do not. I don't know. So the bleaching event was the worst. It was worse south. So I think it depends a little bit on how far south Andros is. It might have been spared because these northern reefs, it wasn't as bad in like Broward County and moving up West Palm Beach County. It was really, really bad down in the Keys. That was the worst of it, was down in the Keys. Yeah.
So, related to that, we'll grab another question from our Zoom audience. Are some corals more heat tolerant than others?
Yes. Some species of corals are more heat tolerant, and actually some individual corals are more heat tolerant. So there'll be cases where you might look out at a reef and you have the same coral species, two colonies side by side, and one bleaches and one doesn't. And so again, that's another area of research that's going on to see what the differences are in those corals, and can we use that information to help create corals that would be more thermally tolerant. The other thing is that some of the algae that live inside the corals are more tolerant of heat than others. So that's another really big area of research, is looking at can we kind of convince the corals to take in the algae that are more heat tolerant, and maybe can do that when they're babies. As it's harder to get them to switch once they're older, but if we give them the heat tolerant algae when they're young, will they keep them? Can we use that to somehow help them withstand these temperatures? Yes, Zach?
So a couple of other questions from our Zoom audience. We've heard a lot about microplastics, and sometimes we hear about sunscreen and the varying realities of the impacts of these things. So one person asks, are plastics in the ocean affecting coral reefs? And then somebody else asks a question about the impacts of chemicals and sunscreens. Are they really having a big impact on reefs overall?
I think my kind of overall answer to that would be that climate change is the biggest factor. Right, so increased temperatures, that's just a really, really huge, devastating thing. And then also ocean acidification, which is basically the lowering of the pH of the ocean because of when you have more CO2 in the atmosphere, more of it gets absorbed by the ocean, and what that does in the ocean is cause a lower pH. And corals build their skeleton out of calcium carbonate, and that change in pH affects their ability to do that. So those sort of global stressors that are happening worldwide, I would say, are the biggest threat. However, those are very hard to address, right? Like, it's very hard to slow this train, and I think we're trying, and more people are understanding that we need to try to do something about climate change. But it's just a very slow thing to reverse. In the meantime, if we want our coral reefs to be able to withstand these stressors that are coming with climate change that we might not be able to address immediately, we need to address local stressors. And that's things like nutrient inputs from runoff, plastic pollution, sunscreen. Those sorts of things are probably less impactful, but the corals can't handle all the stressors on top of each other. So we got to try to deal with the things that we can in an immediate sense while we work on those longer term solutions as well. I did put a flyer in the back that's, I think I got that from NOAA. That's just a sort of like, what can you do in terms of little things that you can do to help the coral reefs. Over here.
How effective or ineffective is beach renourishment after they dredge the sand, put it on the beach, and it all gets washed back out on the shore?
So the question was, how effective, if at all, is beach renourishment? So after, I guess, once there's the beach is eroded away and they bring it back in. Honestly, that's not my area. I don't know much about that. And I haven't really studied that at all. Here?
I recently read an article where they were doing some experimental electrical stimulation of the coral reef with floating solar panels. And I wondered what that was doing. It seemed to be having a positive response, but they don't know why.
You're digging into my brain. Okay, so the question was that he had recently read an article where they were doing some electrical stimulation of the coral reef and using solar panels to provide that electricity. And I'm trying to dig into my brain because I've heard that as well, and I'm trying to remember what it was doing. And I don't want to say and give you the wrong answer. I feel like it was one of the things that we talked about as a group for this Reef Fence project. I think it might have had to do with calcification, so helping the corals with their calcification, but I'm not 100% sure. Yes?
Is there any risk that the artificial reefs become colonized by bad pathogens?
The question was, is there any risk that the artificial reefs will become colonized by bad pathogens? So first of all, I would say there's always a risk, right, of anything like that. I'm not so concerned about pathogens as much as in our area, one of our major issues on our Florida reefs is that they just get overgrown by turf algae and other kinds of little filamentous algae and things that outcompete the corals, make it a bad place for coral babies to survive. So when they try to attach, they're not able to. So I think that's one of our big challenges, is making sure that on our reef that we develop, we have enough strategies to keep the reef from being taken over by different types of algae and turf algae and other organisms that would outcompete the corals. I don't think there's a high risk of necessarily causing some sort of pathogen outbreak in that system. Yes.
One of our Zoom attendees wants to know how long corals typically live for.
Oh my gosh. I want to start by saying I did my PhD on algae. So how long do corals typically live for? I am not going to give you a very precise response on this because I honestly don't know an exact response, but it can be hundreds of years. You can have these huge, huge colonies that are hundreds of years old. Yes?
Is this emerging strategy or developing methodology to use the tetrabromo pyrrole, the TBH? Is that being used currently on both artificial reef building and or dead reefs, say calcium carbonate shells of dead reefs to try to bring them back to life or start growth happening again?
Yeah, so the question is TBP, the tetrabromo pyrrole, is that being currently used on any reefs or artificial structures to enhance coral recruitment? And the answer to that is no. We're just starting to even consider that and looking at, you have to think about, before you put anything out into the ocean, you have to think about the other consequences, right? Like I told you, it doesn't just induce settlement, but it's also antibiotic. It has other impacts on other organisms. And so we're doing a lot of looking into, what kind of effects does it have long-term on the corals that are settling with it. In fact, we're finding some really interesting evidence that it enhances survival in corals. So when we settle them with TBP as opposed to on a natural surface, we actually get higher survival. That's very preliminary. We have to get more replication to make sure that that's really happening, but we have some really interesting results about that. But yeah, it's before we transfer anything to actually into the field, we want to make sure that it's really safe and that that's okay to do. One of the kind of late-breaking investigations last year so also I should say corals spawn once a year. So this is a field where you're very limited in your ability to run these tests. But last summer, one of the very last things I tried with like the very stragglers of the corals that we had left to try was pre-treating the corals with TBP. So giving the corals TBP, rinsing them so that there was no more TBP in the water, and then putting them in a dish to see if they would still settle, and they did, which could be huge for application because then we could treat the larvae, rinse away the TBP, put the larvae out, and they would still settle. And so we're not introducing that extra thing into the environment. Yeah.
All right, Jenny, I've got kind of a tough evolution question from
geez.
our Zoom audience.
I also went to school like 12 years ago or something.
How is it that corals have evolved to need algae to live? Wouldn't they first have developed the ability to live alone and not symbiotically? Is there just not enough food for them to feed on without the algae?
Okay, whew. Yeah, that's a good question, and I'm gonna like skim it here. So I don't know the exact like evolutionary origin of corals, but what I will say is that coral reefs exist in what we call an oligotrophic environment. So it's basically an environment that is very nutrient poor. So in order for them to live in that nutrient poor environment, they rely on those algae that live inside of them to help provide them with food and do that photosynthesis. So that's part of the very like complex ways that coral reefs even have a functioning ecosystem in the environment that they're in. I don't know if that was a satisfactory answer, but yes.
I have another one, okay.
Oh gosh. Start asking about Mendelian genetics. Let's see. No, go ahead.
We have the discharges.
Yes.
And we get the blue-green algae, which is cyanobacteria. Wouldn't that help them?
Okay, so the question was, when we get the if we get the discharges here and we are getting cyanobacterial blooms, blue-green algae blooms, would that help the corals? And the answer is no. So the algae that live inside the corals are very specific algae. So it's not just any algae, it's like very specific algae that live within the corals. It's a specific species.
Any other questions from the Blake?
Symbiotic relationship between the microbes and bacteria and the host. Do both organisms produce the same chemical, which we think are important? Both, like, in the human case, the butyric acid, that's produced by man as well as the bacteria.
So the question is, in a symbiotic relationship with a microbe and a host, do both the microbe and the host produce the same compounds? That's the question. I would say it depends on the compounds. Some do, some don't. So some are exclusively produced by either the symbiont or the host, and then there's some that, some compounds that are produced by both symbionts and hosts.
Particularly interested in the ones that we think are important in terms of regulating helping both parties. Like, for instance, the tetrabromo pearl, is that produced by the coral as well?
Okay, so the question is the tetrabromoperol produced by both the coral and the bacteria? The answer is no, it's only produced by bacteria. Yeah, the reason, so the reason the bacteria are producing it is because it's an antibiotic. So if you think about bacteria, I told you there's like 5 million bacteria in a teaspoon of seawater. If you look at a biofilm on a surface, there's even a higher density of bacteria. So those bacteria are constantly fighting with each other for space and nutrients. And so that's why a lot of bacteria produce antibiotics, because they're competing with each other constantly in their microenvironment to, get that space, get those nutrients that they need, and that sort of thing. And so it's sort of like a lot of things, I was telling you that sometimes we're looking for drugs in these different organisms. Well, those organisms aren't producing those compounds so that we can have an anti-cancer drug, right? It's doing something for the organism, we're just using it. And it's the same with the corals, I think. It's, the bacteria is producing this compound to help it live in its environment, but it's helpful somehow to the corals, so the corals have learned to recognize it. My guess would be that, it's an antibiotic. Maybe it provides an environment for this baby coral where it's safer from infection. So it's just, over evolutionary time, realized that if it settles near this compound, it's going to have a greater chance of survival.
And I think we'll wrap up with one last easy question from our Zoom audience. At what stage, I think you've got this one, at what stage in the coral's life cycle do the polyps start to absorb symbiotic algae?
Okay, okay. Now we're going to go through a little extra primer on coral reproduction. So corals actually reproduce in two different ways. We have what we call brooding corals and spawning corals. Brooding corals take sperm in from the environment and have internal fertilization inside the coral, just kind of like we do. And then they release those fully developed larvae. So what they release into the water is actually that swimming little blob. Spawning corals actually release their eggs and sperm into the water. That egg and sperm move into a big soup in the water, and fertilization happens in the water, and then the larvae develop in the water, and then they move down. So, all that to say, the symbiotic algae in a brooding coral is typically passed along from the parent to the offspring. Because all that fertilization, everything's happening internally, they get their algae when they're in their parent, and then they're released with their symbiotic algae. Spawning corals do not have their algae typically until after they settle. So when they settle, they're clear, and then in the next few weeks to months, if the algae are available in the environment, they'll start taking them in and start getting their symbiotic algae.
All right, folks, help me give one more big round of applause to Dr. Sneed. So thank you all for sticking around. We had a long Q&A session tonight, but that means you guys are interested, and I love when the questions go on and on. Unfortunately, the library eventually kicks us out. Just a reminder, 1 week from tonight, I'll be doing that fun presentation on all the weird ways that fish court and reproduce. This Saturday is our Ocean Protectors Day out at the Florida Oceanographic Coastal Center. And most importantly, on your way out tonight, on the back table, pull out your phone. open up your camera, take a picture of that little QR code, and sign up for our advocacy updates. We're not going to bombard you with a lot of information, but when something important happens, like freshwater discharges start, or there's a bad piece of legislation that we need you to speak out against, that's going to be the best way for Florida Oceanographic Society to get that info into your hands. Folks, thank you again for making this series such a success, and we'll see you next week. Bye, everybody at home.